Hello, everyone. In this lecture, I'll be reviewing the various activities of microglia and how they support a healthy nervous system. I'll go over how they're activated and how they construct the inflammasome to mediate inflammation. And lastly, I'll cover this fantastic paper by Madry et al. Titled Microglial Ramification, Surveillance, and Interleukin-1 Beta Release are regulated by the two-pore domain potassium channel, THIC1. I hope you enjoy. So microglia are the macrophages of the brain, and they broadly function to destroy pathogens and scavenge dead or dying cells. And they do this through the process of phagocytosis, whereby they envelop their substrate, and the resulting internal vesicles are then merged with lysosomes, and they form those phagolysosomes, where the contents are degraded. So microglia can exist in three general states or phenotypes. Although I should stress that these are very broad, and technically there's more of like a continuum of activation, and some types of activated microglia can have completely distinct substrates compared to other types of activated microglia. For example, there's probably a mode or a setting in microglia for synaptic pruning, and maybe another one for bacteria, another one for dead cells, etc. So just keep that in mind as we go through this entire paper. But nevertheless, the first general type of microglia are the ramified or resting microglia that are very actively sensing the local environment and facilitating synaptic communication using these very long extended processes, which kind of resemble dendrites. So it would be a misnomer to think of non-activated microglia as laying there dormant, because they are in fact highly active, and they're essentially using their dendrite-looking protrusions to communicate and communicate with synapses and neurons, and they make sure they're healthy and functioning properly. Ramified microglia are marked by the expression of IL-10, interleukin-10, and TGF-beta, tumor growth factor beta. These are two very important anti-inflammatory signaling proteins. So if you were doing, for example, an immunohistochemical stain for ramified bacteria or ramified microglia, you would use the IL-10 or TGF-beta antibody. And we'll discuss these two proteins in a moment. Another phenotype is reactive or activated microglia. They are amoeboid, amoeboid in appearance. They look like amoebas, and they're highly motile. And they're also actively searching for phagocytic substrate like dead cells or protein aggregates or pathogens. These reactive microglia are spherical, so they look like amoebas, and they're crawling around. And they use various cues or chemoattractants to home in or locate cellular debris or viruses, for example. Reactive microglia are distinguished by the expression of IL-1 beta, and I have a slide coming up on IL-1 beta. But in general, we can say that IL-1 beta is the most classic marker of inflammation, alongside with TNF-alpha, tumor necrosis factor alpha. So these are the antibodies that you would probably use if you want to look for reactive microglia in the brain. And lastly, phagocytic microglia are the last class of microglia, and they are also amoeboid in appearance, but they are actively phagocytosing substrate. And often they have the corpses of dead cells inside their lumen being actively degraded in autophagosomes. And a good reason to be interested in this topic, microglial dysfunction is actually a major area of research in Alzheimer's disease and many other neurodegenerative disorders. So microglia constitute upwards of 10 to 15% of the total cells in the brain. And these same cells have quite the responsibility as we age. And we get older, as we get older, our DNA begins to mutate, our proteins begin to misfold, our cells just generally, they don't work as well. And when our cells die or our proteins misfold in insoluble plaques, it's the job of the microglia to clear them out. And the failure to clear out the debris leads to a vicious cycle of inflammation and cell death, and it can just spiral out of control. On the other hand, if microglia are too eager to phagocytosed material, perhaps due to the presence of age-related pro-inflammatory molecules, such as amyloid beta 42, they can become deleterious in phagocytosed living neurons or active synapses or astrocytes. So it's actually really a delicate balance, and it's kind of amazing that these microglia behave for most of our life, and perhaps not surprising that they become dysfunctional as we reach older age. Amyloid beta plaques are potent activators of microglia, which can result in collateral damage, such as excessive neuron and synapse phagocytosis. So again, amyloid beta is the pathological peptide in Alzheimer's disease, and it's been proven to activate microglia. So this is a very hot area of research right now, and I think that makes it more exciting to give this presentation. So let's get caught up on some of the basics. So we're going to begin with our microglia. This will be our microglia. It's going to be pretty large. And in this slide, we're going to talk about how the microglia interact with different parts of other cells, such as a synapse. Let's draw say this is a neuronal cell body down there. And let's say this is an astrocyte over here. So we're going to talk about how microglia interact with each of these different structures. So synapse. Synapse, we know synapses have lots of receptors. So these are different receptors on the synaptic membrane. And these receptors are glycosylated, right? They have these carbohydrates, these sugar chains attached. And these sugar chains that are attached are called sialic acids. And sialic acids actually function as a don't eat me receptor for microglia. Microglia have a specific receptor that binds to sialic acids and is called the cyclic 11 receptor. And so cyclic 11 binds to these sialic acids. When it does that, I'm not sure of the mechanism, but it inhibits phagocytosis. So basically, synapses that are healthy have these sialic acids that protect them from phagocytosis from microglia. However, in a process called synaptic pruning occurs most frequently during adolescence, and there's evidence it occurs during neurodegenerative diseases. In these cases, what can happen is a certain enzyme gets released by the microglia called neuraminidase. 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 Let me fix that. Neuraminidase, and it functions to. It's a glycosylase that will go and start chewing up these sialic acids. It's called desialylation. It's not a fun word to say. Desialylation. And these desialylated receptors lacking these sialic acid sugars are actually recognized by the complement system. Complement system kind of resembles the antibody system, but essentially what happens is neurons can release this protein called C1q. It's a complement protein that binds to desialylated receptors on the synapse. And these C1q proteins are very small. They get released by the microglia and they diffuse through the extracellular matrix. And if they find a desialylated synapse, it will bind to that receptor. And C1q itself is a receptor for this protein that's called C3. C3 binds to C1q that has bound to a desialylated synapse receptor. And this, when C1q binds to C1q, it recruits a protein called DAP12 DAP12 is an adapter that recruits a bunch of proteins leading to phagocytosis. So neuraminidase gets released, it cleaves up, it chews up these sialic acids, and this allows C1q to bind to the desialylated receptors, which can then bind C3, the receptors on microglia, and through this DAP12 adapter, it initiates phagocytosis. I'm not exactly sure the particular pathway at this point, but that's the general pathway that we're discussing. And this is the process of synaptic pruning. This is how synapses are selectively eliminated by microglia, is they recognize desialylated receptors on the synapse. Neurons, say this is a neuronal cell body or something. Neuronal cell bodies express two main don't eat me proteins. One don't eat me protein is called CD47, very important. The other one is called CD200. It expresses these two proteins called CD47 and CD200. And these are called so-called don't eat me signals because they bind to receptors on microglia and basically inhibit phagocytosis. On the microglia, we have, for example, CD200R. CD200R is the receptor for CD200, and the receptor for CD47 is SERP alpha, or SERP A, I'll call it here. These guys can bind to each other. And this binding, broadly, it has anti-inflammatory properties. It inhibits phagocytosis. Well, I'll go into a little more detail with the CD200 because I'm familiar with its pathway. CD200, when it binds to CD200 receptor, when it binds to CD200, recruits an adapter, like many proteins do. It's a tyrosine phosphatase, receptor tyrosine kinase. So it autophosphorylates itself, and it produces a binding domain for this DOCK2. It's a docking. I think of it as like a docking protein because it binds to it and it docks to autophosphorylated CD200 receptors. And DOCK2 recruits a RAS gap, guanine activating protein. So what does guanine activating protein do? It activates the GTP of RAS. So it causes RAS to drop its GTP in exchange, and it ends up with GDP, guanine diphosphate. And guanine diphosphate, RAS normally activates phagocytosis and inflammatory conditions in general. But since RAS gap is inhibiting RAS, we don't see activation of phagocytosis. So through that mechanism, we can say CD200 inhibits phagocytosis because it's inhibiting RAS. We see something kind of similar. I'll go into a little bit of detail with the SERP alpha receptor. SERP alpha, when it binds to CD47, it recruits a tyrosine phosphatase called SHP2. SH2. It's basically a phosphatase. It's a tyrosine phosphatase that inhibits various pro-inflammatory signaling pathways by cleaving phosphates off. And I don't think that specific targets have been identified, but it broadly functions to inhibit phagocytosis. So these are the two main don't eat me signals on neurons. It's CD47 and CD200. And we also have anti-inflammatory pathways being promoted by astrocytes. Astrocytes can help function to dampen the inflammation of any particular tissue, or obviously brain tissue. And it does this through IL-10 and TGF-beta. IL-10 and TGF-beta are the classic anti-inflammatory signaling proteins, and they can be released by astrocytes, and they bind to receptors on microglia. They are not, they don't have an interesting name or anything. It's just IL-10R for receptor and TGF-beta receptor, R for receptor. And these guys signal through slightly different pathways. The TGF-beta receptors signal through SMADs, which I do have a lecture on if you're interested in, and they broadly function to, so if that was inflammation, SMADs generally inhibit inflammation. And in IL-10, when it binds to IL-10's receptor, it signals through the JAK-STAT pathway. JAK-STAT involves, so when IL-10, it binds to IL-10 receptor, it causes the receptor to autophosphorylate itself, which produces binding sites for these JAK kinases, which will then dimerize. And the JAK kinases that have dimerized are able to phosphorylate and activate the STAT pathway, such as STAT3. And STAT3 can inhibit inflammation. And again, some of these pathways are kind of general because I'm sure you can find a paper somewhere that says STAT3 promotes inflammation. It all is in context. But IL-10 and TGF-beta generally inhibit inflammation, and they do that through in IL-10's case, they do it through JAK-STAT signaling. JAK right here, STAT, and they inhibit inflammation because STAT is a transcription factor. SMAD is also a transcription factor that inhibits inflammation. And there's one more important player that I wanted to mention is TREM2. TREM2, I forget what it stands for, but it's a really important receptor that has been, some certain mutations in TREM2 have been linked to Alzheimer's disease and other neurodegenerative diseases. And what they found is that these mutations in TREM prevent or basically cause the microglia to be rendered immobile. They are not able to search and find phagocytic substrate. And so TREM is a receptor that binds to an unknown eat me signal. There is some evidence that it binds to amyloid beta 42, but it's still being investigated. TREM2 in some way leads to phagocytosis of its substrate, and it does this also through the DAP12 adapter, as we see over here with C3. And so DAP12 somehow promotes phagocytosis through TREM2, and TREM2 is interesting because it's implicated in Alzheimer's disease, although not very much is known about it. It is known to activate the TREM2 is known to activate the mTORC signaling pathway, and thus it can help control metabolism. This is a very recent paper that found TREM2 to influence the mTORC pathway, and through the mTORC pathway it can influence metabolism. Metabolism specifically of microglia, which might be very interesting. So there's a lot to digest in this slide. Let's go through it one by one. So we have our synaptic pruning events. Basically, these receptors lose their sialic acid groups due to the action of neuraminidase. Neuraminidase can be released by microglia where it starts cleaving the sialic acids. And when sialic acid has been cleaved, C1q, also released by microglia, can bind to these desialylated receptors. C1q is a receptor for C3, C3, when it binds to C1q, can recruit this adapter, DAP12, and through DAP12, it promotes phagocytosis or synaptic pruning. On the other hand, if the sialic acids are still present on the receptors, they can bind to Siglic. Siglic 11 can bind to sialic acids, and it inhibits phagocytosis. Not sure the pathway, but it does. Neurons express copious amounts of CD47. CD47 is a don't eat me receptor that binds to SERP alpha on the microglia. SERP alpha activates a tyrosine phosphatase, SHP2, which inhibits phagocytosis or inflammation in general. Neurons also express CD200. CD200 signals or binds to CD200 receptor, which recruits DOC12, the adapter, which recruits RAS-GAP. RAS-GAP hydrolyzes the GTP of RAS, thus inhibiting it. And since RAS is involved in phagocytosis, we could say CD200 inhibits phagocytosis. And lastly, we have two anti-inflammatory cytokines that get released by astrocytes, includes most notably IL-10 and TGF-beta. And they signal through the SMAD and the STAT3 signaling pathways, ultimately inhibiting inflammation. So if you have any questions about this slide, feel free to ask, but these are a couple of the really important pathways that are currently being investigated in neurodegenerative disorders. In this slide, we're going to talk about the construction of the inflammasome. The inflammasome is the multi-subunit complex that facilitates inflammation, primarily through the release of IL-1 beta. We'll get to that in a second. But first, let's imagine this is our extracellular matrix, and this is our microglial cell. And let's see. Let's look at the most important ways that inflammation begins. So let's discuss three important receptors. One very important receptor that you've probably heard of, tumor necrosis factor alpha, or R. There should be an R there. Let's add an R for receptor. We also have our toll-like receptors. There's lots of different kinds of toll-like receptors. And we have IL-1 beta receptors. IL-1 beta receptors. So IL-1 beta receptors bind IL-1 beta. Toll-like receptors bind to PAMPs, pathogen-associated molecular pattern, which is basically from microbes. Microbes can activate toll-like receptors. And what's fascinating about all these different inflammation sensing receptors is they tend to converge on a single pathway. And this pathway begins with IKK. IKK stands for inhibitor of kappa beta kinase. Inhibitor of kappa beta kinase. So the inhibitor of kappa beta kinase, as the name suggests, is a kinase that targets the inhibitor of kappa beta, or I-kappa beta, inhibitor of NF-kappa beta. So it inhibits this protein, inhibitor of kappa beta. And what does the inhibitor of kappa beta do? It inhibits NF-kappa beta. NF-kappa beta you might recognize as a really important player in inflammation. It definitely is. So this is a double negative. The inhibitor of kappa beta kinase phosphorylates this protein, the inhibitor of kappa beta, and this inhibits NF-kappa beta, or leads to the activation of NF-kappa beta. So if you look through this double negative, IKK can be said as activating NF-kappa beta. You just have to see through the double negative. If we go back to this phosphorylation that was placed on the inhibitor of kappa beta, when it gets phosphorylated by IKK, it recruits the proteasome. It causes proteasomal degradation. So the phosphorylation of the inhibitor of kappa beta by IKK, or the inhibitor of kappa beta kinase, causes proteasomal degradation. And this frees up NF kappa beta. It releases NF kappa beta from the chains of the inhibitor of kappa beta, and NF kappa beta translocates to the nucleus. This will be our nucleus. NF kappa beta is a transcription factor that will move to the nucleus and influence gene expression. I won't go into the specifics. I'll just say NF kappa beta gene expression. It upregulates proinflammatory proteins. Some of the really important pro-inflammatory proteins that are upregulated include pro-IL-1 beta and NLRP3. NLRP3 is the primary subunit of the inflammasome. So NF kappa beta is upregulating the core subunit of the inflammasome. In ILRP3, the inflammasome should remind you of the apoptosome, because they both function in similar ways in that they oligomerize. So if you remember the apoptosome, it oligomerizes to form that APAF1 complex, which then recruits the caspases, not caspase, NLRP3. Well, in a similar way, NLRP3 is also oligomerizing. However, it doesn't just do this on its own. It needs various activation cues. So some of the things that activate the oligomerization of the inflammasome subunit NLRP3 includes potassium efflux, the movement of potassium out of the cell. So potassium in some way prevents the oligomerization of the NLRP3 inflammasome. And potassium is ejected out of the cell through these P2Y receptors. P2Y you might recognize as an ATP receptor, which we'll talk about on the next slide. But P2Y is a receptor for ATP. When ATP binds to P2Y, it opens up a channel causing the efflux of potassium. So potassium is being squirted out of the cell when ATP binds to P2Y. And we'll talk in the next slide how ATP is a find me signal. It basically it's actually a pro-inflammatory cytokine, because ATP typically is only released when a cell ruptures or is dying. ATP gets released, binds to this P2Y, which is a G protein coupled protein. Anyways, we'll talk about that in a second. And it leads to the efflux of potassium. And this loss of potassium activates the oligomerization of NLRP1 or 3. Other things to activate the oligomerization of NLRP3 includes calcium, very importantly, and reactive oxygen species. So calcium flux and reactive oxygen species both activate the oligomerization of NLRP3. So we keep talking about this inflammasome, but what does it actually do? How does it cause inflammation? And that's going to be the second step. So after the NLRP3 proteins have oligomerized, I think there's 7 that oligomerize in total. After they've oligomerized, they produce a binding site for this protein called ASC. And when ASC is bound to the oligomerized NLRP3 inflammasome, it has what's called a CARD domain, C-A-R-D, and that stands for caspase recruitment domain. And the caspase recruitment domain is specific for a certain caspase. The caspase that it actually recruits is called caspase 1. And specifically, it's pro-caspase-1 because it's not in its active form normally. So pro-caspase-1, kind of running out of room. Pro-caspase-1 gets recruited to ASC and gets cleaved into an active form. So we get active caspase-1. So the inflammasome's main function is to recruit this ASC protein, which has a card domain, to activate caspase-1. So what does caspase-1 do? It cleaves IL-1 beta into an active form. So instead of being pro-IL-1 beta, is now IL-1 beta. IL-1 beta can be secreted and generates the inflammatory environment. It causes inflammatory conditions. Because look over here. We have the IL-1 beta receptor inducing the expression of NF-kappa beta, which produces more inflammation. So just to summarize, we have all these different pro-inflammatory receptors, toll-like receptors, tumor necrosis factor receptors, which binds tumor necrosis factor. They lead to the activation of the inhibitor of kappa beta kinase. When the inhibitor of kappa beta kinase phosphorylates the inhibitor of NF-kappa beta, right here, it gets degraded. This relieves the inhibition on NF-kappa beta, so it can now translocate to the nucleus, influences gene expression. Some of the expression is things like IL-1 beta and the inflammasome subunits. The inflammasome is coerced to oligomerize when potassium is expelled, is effluxed throughout the cell, out the cell. And other things that strongly activate inflammasome oligomerization include calcium. Calcium transients are very important, as are reactive oxygen species. When the inflammasome has oligomerized, it recruits this ASC, which contains the caspase recruitment domain, recruits pro-caspase-1, cleaves it into active caspase-1. Active caspase-1 then induces inflammation by cleaving pro-IL-1 beta into active IL-1 beta, which is then secreted, leading to the inflammatory environment of the tissue. Okay, so let's tackle this paper titled Microglial Ramifications, Surveillance, and Interleukin-1 beta release are regulated by the two-pore domain potassium channel THIC1. So potassium is the primary ion responsible for regulating microglial membrane resting potential. So this is really interesting, especially since we just discussed how the inflammasome is principally triggered by the efflux of potassium. And potassium efflux mediates membrane hyperpolarization because if potassium is leaving the cell, it's positively charged. So this causes hyperpolarization. This hyperpolarization is known to activate microglia. So the activation of microglia is marked by the hyperpolarization of membrane potential from roughly negative 40 millivolts, and it usually ends up around, it gets hyperpolarized to around minus 70 millivolts. Microglia are known to be activated by ATP and ADP, which are released from dead cells. But the mechanistic relationship between ATP and potassium efflux is not really very well understood. So the idea here is that since ATP can activate microglia, and activated microglia are marked by potassium efflux and hyperpolarization, the authors wondered if they wanted to know how ATP led to potassium efflux. And so that's going to be the idea of this paper, is investigating that relationship. And this is what they discovered. So the G protein coupled ATP receptor, P2Y12, the ATP receptor P2Y12 opens up the outward rectifying potassium channel, THIC1, and this causes membrane hyperpolarization. So the primary ATP-ADP receptor on microglia is this P2Y12 receptor. So P2Y is the class. It's just in there's 12 specific homologs. These P2Y12 receptors activate THIC1, outward rectifying potassium channels, meaning potassium flows out of the cell. They're outward rectifying. So they mediate the potassium efflux. And P2Y receptors do this through a G-coupled protein receptor mechanism because when the G protein activity was blocked using a small molecule, potassium efflux in response to ATP was blocked. THIC1 activation by P2Y12 is required for inflammasome formation and IL-1 beta release. So this ATP P2Y THIC1 potassium pathway is required for inflammasome formation and IL-1 beta release. And in my mind, this was the big discovery of the paper. I think this was the most fascinating part. And if you think back to our last slide, it's really important because it means that perhaps calcium in reactive oxygen species might be downstream or less important. But the main thing is the hyperpolarization, the release of potassium, and this facilitates the oligomerization of NLRP3 and the activation of caspase-1 and the release of IL-1 beta. However, this paper also discovers that tonic baseline THIC1 activity is required for microglial surveillance, but not motility. So this might be a little bit confusing. So low levels of THIC1 potassium efflux occurs constantly. It's this tonic, low-level activity of THIC1 that is required for proper microglial surveillance and the ramified phenotype. And although massive THIC1 mediated potassium efflux triggers IL-1 beta and inflammasome formation, it does not actually affect motility. So you can have activation in the absence of motility. So potassium efflux, although it may be activating the cells when it's a massive release of potassium, it's not actually required for the movement of microglia. That's operating under a different set of pathways. So let's get into, let's see if I can depict this suggested pathway in the next slide. Okay, so this is going to be our microglial membrane. And the idea is that they're adding ATP to these microglia. They're activating them with ATP. How does that happen? So as I mentioned, it occurs through that P2Y12 G-coupled protein receptor. Except ATP doesn't directly bind to P2Y12. It actually binds to a subunit that is on the outside of the membrane called CD39. CD39. CD39 is an ATPase, and it actually hydrolyzes the ATP or the ADP into ADP or AMP, depending on which it's binding. And remember that this ATP is derived from dead or dying cells, right? They release these find me signals in the form of ATP. ATP is an inflammatory cytokine. And it diffuses through the extracellular matrix. It encounters CD39, which is attached to P2Y12. And the hydrolysis of ATP into ADP or ADP into AMP, either or, activates the beta and the gamma subunits of the G-coupled protein, the P2Y G-coupled protein. These are the subunits of the G protein that get activated when CD39 hydrolyzes as ATP. And the beta gamma subunits diffuse to the THIC1 potassium channels. The THIC1 potassium channels are also embedded in the membrane, obviously, and they're outward rectifying potassium channels, which means when they're activated by the beta gamma subunits, it causes the efflux of potassium. So potassium efflux. And before going any further, I want to go over this idea that the -40 is the resting potential of microglia. -40 resting potential is normal business as usual. And in order for it to get there, it's actually kind of regulated through THIC1. THIC1 regulates the tonic baseline activity of the potassium currents, right? And it's important for THIC1 to be semi-active even under baseline basal conditions because it helps maintain this -40 resting potential. This -40 resting potential is what's responsible for the ramified phenotype. So this is pretty important, right? The ramified phenotype is what we observe at a -40 resting potential, which is partially due to a low level efflux of potassium through THIC1. So THIC1 is actually required for the ramified phenotype. However, let's go back to our inflammatory conditions. We see the beta-gamma subunit of P2Y diffusing over and activating THIC1. And instead of saying potassium efflux, I'll say massive potassium efflux. It causes the channel to just burst wide open and cause potassium to flood out of the cell. So this is depicting a flood of potassium leaving the cell because THIC1 goes from low-level business as usual potassium efflux to just a floodgate. It opens up the channel and all the potassium leaves the cell. Maybe not all, but a lot of it does. And this leads to a roughly -70 resting potential. And this is the resting potential that we see in activated microglia. This causes, this is what's called the hyperpolarized phenotype. So we get hyperpolarization. We also get decreased cyclic AMP. We also see calcium flux. We see all these events happening. Some of this might actually be due to other receptors being activated at the same time. Maybe there's LPS or lipopolysaccharides from bacteria that might be activating toll-like receptors. So I'm adding this because this is another way that we see calcium flux or decreased cyclic AMP. So massive potassium efflux causes hyperpolarization, decreased cyclic AMP, calcium flux, and all these things are going to trigger the construction of our inflammasome, right? Our inflammasome is constructed with the NLRP3 subunits. This causes the activation of caspase 1. Caspase 1 cleaves pro-IL-1 beta into active IL-1 beta or mature IL-1 beta, which gets secreted, leads to inflammation. So the big finding of this paper, it has to do with this THIC1. THIC1 is the outward rectifying potassium channel that mediates both the normal resting potential of normal ramified microglia, and it's also responsible for hyperpolarization and inflammasome formation and IL-1 beta secretion. Although interestingly, THIC1 doesn't appear to have anything to do with motility. So motility is operating under a different pathway or something, different cues. So although dead cells are known to activate microglia through this P2Y of CD39 mechanism, it doesn't appear to affect motility directly. So in this slide, I'm going to show you a video from the paper. These are microglia, directed motility in rat hippocampal slices. These are live microglia, and they induce damage using a laser. Right there, that's where the damage is induced using a laser. And here, they are supplying the cells with ATP. You can see that's the pipette in green, releasing ATP. This recruits the microglia. So again, we have a laser inducing damage in a roughly 10 micron location, recruits microglia. And then we see ATP recruiting the microglia as well. In this video, we're seeing microglia in two different conditions. We're seeing these. We're seeing the activation of microglia through the addition of ATP again. We see on the left slide what happens when PBS is administered, or sorry, PSB. PSB is an inhibitor of the P2Y or PY212 ATP receptor. So when ATP cannot bind to P2Y, this is what happens. And we see almost no directed motility. The microglia are pretty much indifferent to the addition of ATP. Over here we see the inhibition of microglia with TPA. TPA inhibits THIC1 channels. We see that the motility is pretty much unaffected. So THIC1 is not needed for motility, but the ATP receptors are needed for motility. In this video, we're seeing the activity of microglia over time in a wild type condition in vivo and a THIC1 knockout condition. And what they find is that the ramified phenotype with all these processes sticking out is vastly inhibited when THIC1 is gone. So THIC1's tonic efflux of potassium is important for the baseline normal phenotype of microglia and their ability to sense their environment and other such things. Anyway, if you guys have any questions about this paper, feel free to ask me. If you have any questions about my presentation, just let me know. And thanks for watching.